Hydrilla was first discovered in 2011 in the Cayuga Inlet. We didn't think it was in the lake. It was in Cascagoula Creek, a little bit of Linderman Creek, which is that little trip that you walk the bridges over the bridges in Cass Park. That's Linderman Creek. So when that happened, there were about 3 weeks of some very exciting, intense meetings with everyone in the room who maybe has authority, maybe has jurisdiction, maybe has any experience with aquatic plants or water resource or land resource management to figure out what to do. This was an unprecedented discovery. Hydrilla had been in New York before. It was in 6 ponds that were isolated, so it was just not being addressed. There wasn't a big concern that it would spread anywhere. This was the first time that hydrilla had been found in a connected flowing water body.
That looks good, full screen.
So the threat was gigantic. It threatened Cayuga Lake, of course, all the Finger Lakes just because of boat traffic, not just downstream, and then the Great Lakes for the same reason. So we all had to get in a room, and it was pretty exciting. So then there are several segments or topic areas that came up that had to be dealt with, and they're still being dealt with today, but less excitingly. Because we've never had an event like this or a plant quite like this, there also hadn't been this large of response launch or trying to do such a rapid response in such a large system. So even that process didn't exist. So the first thing we had to do, we all figured the DEC would take care of it. I know. But that just seemed like a good idea. The DEC would take care of it. What we found out was that even though they had an invasive species program, whenever their authority was given to them, it was for terrestrial plants and animals and aquatic animals. There's one missing. They literally had no legal authority to take the lead. That was very shocking. We had some other issues. Oh, are we up? Yay! Okay, we got through that slide already. Uh-oh. I guess I need an arrow key. How do I move it? Why am I not moving it? Enter? I just need to move it. Hey, the arrow keys aren't working.
Gosh. It's just thinking for a very long time.
Okay. Well, okay, so there was that. So regulatory already that created a good deal of chaos for us. And then what we might have done after that was lean into our regional PRISM. Those are partnerships for regional invasive species management. And there were several of those in the state that were funded and operating. And the Finger Lakes was a defined area, but it was not funded. It didn't exist. So we couldn't lean in at the state level or regionally. So it really did become a local emergency. So we had to sort that out first. Oh, and here are the other issues. So then we had to figure out coordination and funding because that didn't exist for a program like this either. And then we had to do outreach to explain what hydrilla is, why it's especially unpleasant to have, and what the timing was required to actually have an effective response, and also just to build community support around it. And then we also had to develop a treatment plan because, again, that hadn't happened in the state. So we had to lean into experts in other states. Which one, is that one going to work?
Just click if you click this. Yeah.
Oh, yes. Okay, perfect. Okay. All right. So I've already explained lack of state authority. We also ran into, once we kind of had discovered that we were, at least in the first year, because we were trying to get a quick response and knock things back while we made more long-term plans, we were looking at herbicides. And we ran into some other legal issues that were going to hamper being able to treat in a timely manner or use the chemicals we had just because of one word on the label. So a lot of legal issues, which have been addressed, but they were giant hurdles at the beginning. So that's most of that. So to deal with coordinating and developing a management plan, the Hydrilla Task Force was created. And that involved the DEC, of course, because even though they didn't have authority to lead the charge, they're still the regulatory body that does permitting, oversight, and that sort of thing. So they've been with this program since day one. But it had to be people on the ground locally, agencies and municipalities. So the reason I'm standing here is because we thought it was just in the inlet. The city of Ithaca is the jurisdiction, the municipality around that entire area. I happen to get invited to those meetings, and watersheds in my title. So there you go. So the city was represented on the local task force and seen as kind of a lead agency for a while. Soil and Water has been huge throughout this whole process, leading on permitting, signage, water quality monitoring, that sort of thing. But within the local task force, we also had two smaller working groups, one that did outreach and one that developed treatment plans year to year. So I'll get into that more quickly. Moving on to funding, though, there was no funding dedicated for this kind of effort. So in sort of an emergency situation, some state monies were moved to help the first couple treatments happen. The U.S. Fish and Wildlife Service reallocated funding that had been dedicated to a different program. And they funded this for a number of years. State parks came up with $60,000 for a number of years, just new money. Bridge loans through the city of Ithaca, because grants are usually reimbursement. So Soil and Water was having to do the outlays and then get reimbursed. So the city of Ithaca would do the outlay. And then there were new federal funds that went through Army Corps of Engineers through a research designation, and they might speak to that later. None of these funds were developed for this kind of long-term sustained program, so they haven't stayed permanent because they've drifted back to their original purpose. And then again, like thousands of hours from staff. And then to the outreach and management, these things had to happen in tandem. You can't do outreach if you don't know what your management plan is, but you can't get a management plan accepted if you haven't done outreach to make people aware and understand it. So there was just a lot happening all at once. We've had two kinds of principles from the beginning. One is that we want transparency. So we had just tons of public meetings where we were hearing concerns. We'd go back, we'd try and get the answers. And we also wanted to lead for the management plan with science. First, with the biology of the plant. What makes the most sense? What's the most effective? What's the best timing? What's the least damaging outside of the plant? But your best answer there might not be possible because only some things are legal. Different states have different options that are available. They're not all available here. Some things haven't been technically developed yet, like biocontrol. So you had to figure out what your constraints were. And then when you came to a decision, we would send that out to regional experts in aquatic invasive plant management, and we'd get it peer reviewed. So when the treatment was ongoing, the things that we did to try and optimize the treatment effectiveness and maintain community transparency was monitoring, the typical monitoring. Are you maintaining compliance with your permit? But are you also hitting your targets so that you're being effective with your treatment and not being kind of sloppy about dosing and putting chemicals in without being effective? We monitored the plants, the biomass in the tubers, so that we would see what our trajectory was for removing it or suppressing it, and be able to adjust our treatments. We also monitored native plants so that we could see if there were negative impacts outside of hydrilla, and if there were, what were they? And of course, we monitor water quality for both environmental and public health reasons. The water quality monitoring was designed every year and overseen by the Department of Health, and the results are posted on their website. At the end of the year, for a number of years, these giant reports were prepared and they were available on our website. There's still data and it's available and shared at meetings like this at the end of the year. Well, and there I have this community conference series right there. So that's mostly this slide. So just for changes, I go away. Okay. So there have been some shifts in roles over time. That's partly because of where the funding's coming from, but mostly because over all these years, DEC has grown their capacity a lot, and they've taken on more of a role in leading this. And Army Corps has taken on a much bigger role during some of the years, and that was probably partly some of the funding that the local task force is using was declining. But we're all still at the table. We just have slightly different roles. The local task force folks, we're now more your representative. So we take your voice to the table in helping to make the plan and give feedback about questions that we might want answered, new concerns. And I'll bring that up again. I probably didn't stress that at the beginning. It's been 13 years. We did a lot of work to do our very best job at the beginning, but it's not quite the same world. Maybe the options have changed a little. Maybe they've expanded. Maybe there are new concerns. So it's part of why we're giving you the backdrop and offering again, ask the questions so that we can go try and get the answers. But we still have the same principles. And with that, I'm going to turn it over to Sam from the PRISM that is now funded to talk about their role, some of the achievements and accomplishments that have been made in some areas that are still difficult.
Okay, can everybody hear me? Can you hear me?
Is it on?
Doing just fine.
Can you hear me?
Yes.
Okay, great. All right, thanks, Roxy, and thanks everybody for having me. My name is Sam Beck Anderson. I am the coordinator for the Finger Lakes Partnership for Regional Invasive Species Management. Roxy did a really good job of kind of describing a lot of the partners and the overall kind of climate of this issue over the past 13 years. I'm going to talk specifically about kind of the progression of Hydrilla itself in Cayuga Lake. I'll give a little bit of background about the PRISM itself and kind of where we're situated. And then at the end, I will give a little bit of updates about kind of what our team was able to achieve or some of the achievements that we've had related to Hydrilla this season. Oops. Okay, just 1 second. Okay, so Finger Lakes Institute we are a not-for-profit. We are a department of Hobart and William Smith colleges at the north end of Seneca Lake in Geneva. The Finger Lakes Institute acts as a research institute, translating knowledge and research about the Finger Lakes watershed into collaborative action. Our projects are pretty far-ranging, but overall focus on contaminants, nutrients, watershed management, and of course, invasive species, all focused in the Finger Lakes and surrounding watersheds specifically. The PRISM, so the Partnership for Regional Invasive Species Management, these are Environmental Protection Fund funded programs. The New York State is broken up into 8 different PRISMs. We are the middle one here. We are roughly the size of New Jersey. We have 17 counties. We have some of the larger cities in the state. And this wide range and kind of broad view of kind of what our large area tends to dictate and kind of guide how we're actually addressing invasive species. So we're not out there trying to pull every single piece of Japanese knotweed or every single piece of water chestnut, plants that are spread across the region. What we're really focused in on is leveraging existing partnerships and funding to focus on a common goal, of course, in this situation being invasive species. So what is an invasive species? Of course, it is one that is not native to the ecosystem under consideration, but it isn't just that. An invasive species actually has to have some type of a significant negative effect on the economy, of an area, environmental factors, or human health. So the economy, things like impacts to agriculture, recreation, trade, shipping, tourism, utilities, management costs. Environmental impacts include things like impacts to biodiversity, structural diversity, general natural processes and food, energy dynamics, aesthetics, ecosystem function, services, and more. And of course, human health. In some instances, invasive species can have negative impacts on soil, water, and air quality, flooding, injury, disease, and illness. Things and Hydrilla, lucky for us, kind of has a little bit of everything here. So it, of course, has pretty significant, which I guess we'll get into it here. Regional context in the Finger Lakes is one of the most popular tourism destinations in the state. With our vast array of successful and popular wineries, vineyards, and breweries and restaurants in the Finger Lakes, we bring a ton of people. Of course, also primarily in Cayuga Lake specifically, we have one of the best bass fisheries in the nation. We get thousands and thousands of people spending millions of dollars every single year coming to the Finger Lakes from all over the region, all over the world, to visit our area and utilize our natural resources, specifically our water. Some additional kind of notable facts about the Finger Lakes is that they are hydrologically connected to a number of different large systems. Cayuga and Seneca Lake specifically are connected to Lake Ontario and ultimately to the St. Lawrence River and basically the rest of the world. So there's a lot of things here that make invasive species particularly of interest to us in the Finger Lakes region. But also it makes them, our area specifically, kind of rife for the spread of invasive species as well. So thinking about Hydrilla, one of many invasive species that we focus on at the Finger Lakes PRISM, also known as water thyme. This has a broad habitat range. It has a lot of the similar characteristics of any invasive species. It reproduces very easily, so either by fragmentation, so a small piece of Hydrilla can actually grow into a brand new infestation. It also has tubers and turions, which help it survive for a long time. It can survive over winter and reestablish itself in following years. This is a plant that is visibly serrated, leaves in whorls of 3 to 8. There are a couple of different varieties of Hydrilla throughout the North America. Ours specifically is a monoecious variety of Hydrilla. Threats and impacts. Invasive species of all kinds, like we said, have pretty significant impacts to the environment. So this is a picture of, I think, probably Florida, somewhere in the southeast United States, which actually is suffering from a pretty serious Hydrilla population here. That's causing. This is likely actually mowed. This path here is likely mowed, just literally to use. Just to be able to use this waterway just for its limited purposes here, just maybe a 1 or 2 boats at a time. The Great Lakes Commission of Governors has identified there's been a number of different studies to look at the possible impact of Hydrilla onto a number of different areas economically in the Great Lakes specifically. This, of course, is an estimate and it is a few years old, but just to give you an idea, these are just some of the areas that we're actually interested in worrying about the economic impact of as well. So managing these environmental problems, but also direct economic impact to things like commercial navigation, recreation, tourism, beach use, and angling. So Hydrilla in Cayuga Lake, like Roxy said, it was first discovered in 2011. This initial population was found by the public through the floating classroom. Additional populations were found after that through surveys in other locations. And that general trend has kind of been consistent overall. Like there have been a couple of locations and populations that have been found through active monitoring. But the overwhelming majority of the new populations or just generally the populations of Hydrilla that have been found on Cayuga Lake have been kind of on accident in some ways. So whether it's a member of the public, a scuba diver that just happens to be able to identify Hydrilla, finding it in Sheldrake Point in 2022, or a group of another group of students doing a general rate toss, finding hydrilla in another year. It's pretty hard to find. This is really kind of the needle in the haystack, and a number of all of the different groups that Roxy was mentioning have all taken some part in kind of addressing this, but still it's really hard to pinpoint hydrilla specifically. So this grid shows the progression of hydrilla populations across time, going back to 2011 and up until this year. So basically what we're looking at here is the gray cells are sites not sampled. The blue cells are where hydrilla was not detected, and then the orange cells are where hydrilla was detected. The letters specifically show whether an area was sampled or whether there was some type of treatment, either chemical or mechanical treatment. So a couple of things here to kind of point out to just show how hydrilla is really like a pervasive and really just a tough invasive species to deal with. We're looking at areas like the Ithaca Inlet, where for a number of years we even thought there, despite a lot of surveying and no finds of hydrilla, we're still finding it after a couple of years. And this isn't the only situation that this has happened. We're looking at other locations just outside of the inlet where we're surveying for it after treatment, and we think that it's gone, and then we find it the next year, unfortunately. Even in Lansing and Sheldrake Point, where it hadn't been notified, we're still kind of finding it sometimes a little circuitously. So really what this goes to show is that just there have been a ton of different options or approaches to actually addressing these things, and this just shows the diversity of different activities that have been happening at a number of these different sites throughout Cayuga Lake. Roxy mentioned a lot of these great partners that make up the LTF. Of course, there are more, but these are just some of the main people that have been active in some of these activities specifically. So these groups are really important for just keeping the fight alive in many ways. As other groups have become involved, communication has expanded. So now as new hydrilla populations have spread throughout the lake, we're able to, and things with the DEC have changed, like Roxy said, we're able to sort of expand the network of people that are interested in and kind of able to help and support the mission here. In 2021, the task force created a hydrilla management plan for the lake, which really focuses on adaptive management. And I think that approach is really well observed in this figure here. So really what we're looking at is we're, as a group, I say we're really just a very small part of this whole thing, but where the activities are being done, they're being evaluated and being reestablished. So meetings like this and other meetings like the local task forces helped to support an adaptive approach to managing hydrilla, and it continues to adapt to this day. So like I said, this is just a really brief overview of kind of hydrilla specifically in the lake and kind of some of the activities that have gone on. I mentioned a little bit that we do some of our own work, of course. So the PRISM specifically, our bread and butter really is outreach and education. But we have, had a long history at this point of doing a lot of early detection, rapid response surveys for specifically hydrilla, but also other aquatic invasives as well. So we had a team this summer. We worked very closely with Army Corps of Engineers and the DEC to make sure that we're providing supplementary work and efforts so that no work is duplicative. We're making sure that we're communicating really closely on this work, and it's happening on a weekly basis. So that's for when we were doing treatments up until last year. It was very tightly coordinated there, but also now, even when we're just doing the monitoring efforts, these efforts are really closely coordinated with the partners, which is, I think, a huge strength of the task force. No new populations on our end of the hydrilla were discovered in 2024. You can kind of see some of the locations where we were looking at this year. A lot of these sites were selected somewhat randomly. They're all basically boat launches, but we selected random boat launches and then also went back to some active hydrilla sites to support the monitoring. I mentioned education and outreach. Our program runs a large watercraft steward program, which stations staff around Finger Lakes regional boat launches. Our stewards on Cayuga Lake intercepted hydrilla 3 times this summer. So that really goes to show that kind of integrative approach where we're not just looking at, direct treatment. We're looking at prevention. We're looking at kind of containment. Stewards, like I said, were able to retrieve in May, June, and July. We were actually able to identify hydrilla either entering or exiting the Cayuga Lake. So hopefully these projects and programs will be able to continue to support kind of the general management plan for Cayuga Lake and hydrilla. So that's what I've got. Thanks very much. And I think I'm passing over to either Rich or Emily.
Actually, can we ask questions between the talks?
You can do, Sam, are you still there?
Yes.
Okay.
Yeah, of course. Yep.
Switch the slide. That would be fine. And Rich is going to be up next.
Okay. Well, maybe quickly, because this is
Sam, can you hear the question?
If you speak up, I should be able to hear you.
let me know if you hear me, or do you hear me now, Sam?
Yep.
Okay, so this is current method. Thanks for your talk. One thing that helped me understand also some of where this has come from over the years. So you mentioned that these days it's pretty hard to find the hydrilla, and it's through the rake tosses. I mean, has it always been that way, or can you give me a sense for the largest sort of swell of hydrilla that we've seen in the inlet or the lake? Rossi had shown a picture of the inlet which looked quite green. I don't know how much of that stuff was hydrilla.
That was all hydrilla.
So
that was at the kayak launch at the farmers market. That was actually in Cascadilla right before it enters into the inlet. That was the worst, and also the Cornell Crew boathouse area.
Gotcha.
Yeah, so maybe I can just and by hard to find, I mean when we know where it is, it's not hard to find. It grows like a weed does. So it is where we know it's populated, we can find it. The problem is that it's a really big lake. It's, we all know it, it's over 30 feet long. The liminal or the littoral zone of the lake is massive. We're talking about hundreds and hundreds of acres of area where it could be. And it's really needle in it. That's, I think, where that kind of needle in a haystack really comes from.
Gotcha. No, I'm wondering about in the lake. So that's helpful to know for the inlet, and in the lake that's so deep, I guess. Because I often see these pictures of an entire waterway taken over, and you show a picture from Florida, and that was presumably shallow water, and it was all brown, But I wonder what evidence we have for hydrilla's potential to spread in much colder climes and for water like this.
Yeah, so Rich will get into some of the treatment zones. So hydrilla has really taken hold in some of these areas. I think they're upwards of, I'm not going to misquote it, but many acres are some of these active populations. I will also say that the entire north end and a lot of the south end too of Cayuga Lake is much shallower and warmer than some of the other areas in the middle. So it grows to about 30 feet deep, so around 10 meters. So no, it's not going to grow in, the majority of the lake, but like the parts of the lake that are, many of the parts that are really important are still, susceptible to hydrilla growth. I'm not going to say that it could fill up every single square inch of the littoral zone, but it's already shown pretty clearly that it has the ability. It likes being here, and it will find a place to set up.
Thanks, Sam. Great.
Yep.
So Rich will have some information to add to some of those questions.
Thanks. One more question.
Thanks for your question.
Okay.
It's my understanding that it's very hard to tell the difference between hydrilla and native Elodea. And I'm wondering what is the mechanism you have in place to make sure that the people who are volunteering and all that, and even the stewards, are trained in such a way that they are able to distinguish between the two. Because, it's pretty important if they're identifying it as hydrilla in other parts of the lake, and in fact, it's, Elodea, which is a native plant.
Sam, could you hear that question?
Yes. Yeah. Another good question. It is really similar. We've had people on our staff kind of make the mistake. We go through a pretty rigorous process of identifying new populations when they come up. So when we find it and we know that where it is, we're not as worried about it. But like when a steward finds it, for example, we go through a pretty rigorous process. We even sent, photos of this to the state for confirmation that we were finding, we knew what we were finding. We were confident of what we found, but we, again, have a pretty wide network of people that can help, confirm things. In addition to that, like the staff that we have undergo like a lot of training. So they're trained for about a week. The people that are dedicated staff to this working daily over the summer are trained for about a week on, all of the ups and downs of plant identification. Very specifically, we go based on the lookalike. So we're kind of taking piece by piece, leaf by leaf, and kind of even down to the margins of what the difference between Elodea and Hydrilla are for the folks that are working, on a daily basis doing this. For the stewards, their training isn't as focused at the beginning. They get a 2-day training where we spend several hours looking at invasive species identification processes. And again, that's really when we make extra careful that we know what we're finding. And we train them every other week. We also have kind of follow-up trainings to kind of, a lot of times we'll bring in Hydrilla and kind of quiz people in real time. And, it's an active process, and we're well aware of the differences and the fact that it can be pretty difficult to identify. And we kind of make, every effort to make sure that that's, addressed.
Thank you.
Thanks, Sam. All right. I'm going to hand it over to Richard Ruby.
Thanks. Thanks, Liz. My name is Richard Ruby. I'm a fish biologist with the Army Corps of Engineers. I've actually been involved in this project since we got started helping out with the finds that happened up in Wells College Bay, which that actually Dr. Johnson went out after a class laboratory found Hydrilla, one of the professors there, and notified the network. The task force actually sent Bob up there. Bob actually did a grid and found about 30 acres of Hydrilla. So think of what you saw in the inlet. In Wells College Bay, you saw 30 acres of that. So when we first went out there in 2016 to start doing work, and you were talking about concerns about maybe misidentifying, we go out and do extensive delineations of these patches to determine where the extents of them before we ever decide what we're going to do to actually treat them, whether it's a mechanical application with benthic barriers or whether it's actually. So these patches or these locations that we're talking about have many experts going over them many times to evaluate not only is it there, but how extensive is it? Where is it?
When you say it's over 30 acres, just know what that means. I mean, does that mean that it was like they've taken over those 30 acres and you found nothing else growing there?
Pretty much. In certain areas, it becomes a monoculture.
In the 30 acres.
In that 30 acres, so in the total 30 acres he had, 50% of that was coverage. So it was covered. In other words, you threw a rake and you were going to get Hydrilla. So that means 50% of that. So I misspoke. Of the 30 acres that was there, 15 acres of that was solid Hydrilla growing to the surface, just like you saw in the beginning.
So you say you do the rake toss and like it's all Hydrilla?
Yes. Okay. Yeah. So every point intercept you're throwing, and Bob did it where he actually did 2 rake tosses, drug them out and pulled them in, and then actually did them by. So it is a very extensive plant circle.
Because related to the other question about the native Elodea, I guess if they're coexisting or in some place. But here you're saying that
that was predominantly Hydrilla.
There was nothing subtle about
it. Yeah, there was no doubt.
I see.
You could literally drive up to it with the boat, and it was to the surface.
Okay. Yeah. I don't care.
So that's what we started with. Inlet had that in 2011. They started applications, started treating it, and then it was again found in 2016 out in Wells College Bay, much farther to the north. Getting to one of the concerns that Roxy talked about is the concern with this is that getting out of Cayuga Lake, getting into the canal, and once it gets into the Erie Canal, now it has access to pretty much any water body in the state. So that was the main concern. That's what what our main concern is trying to contain it where it already is. If we can eradicate it where it is, we eradicate it. If not, we want to control it and keep it at those low levels so we don't see those really nasty impacts that can happen when it becomes a monoculture, when it grows to the surface and has all this different. Go ahead, Ben. Next slide. Yeah. Okay, so Roxy talked about really what you know, when we're going out to look at hydrilla. First off, we've identified, and there's been a lot of work to look at the fact that hydrilla is a pretty significant risk to not just the economy, it's also to the environment, or excuse me, the ecology as well. I'll run through some of the different methods that we have to go after this, but if we are doing herbicides, we want to make sure that we're using, we're responsible, use the herbicides as they're approved and at the levels that they're deemed safe. We will also want to make sure that we protect, obviously, Finger Lakes, but we want to protect the bigger Great Lakes area. And there's great concern about these getting in. We treat hydrilla in the western part of the state, out at the Erie Canal and at the Niagara River. Our big concern there is that getting into Lake Ontario and obviously Erie and starting to spread out further. Got a recent discovery now in Chicago area where they have hydrilla. So this is starting to pop up into other areas. And so the concern is to try to keep it contained before it becomes a major impact into this. And as the environment, and we know that climate is changing, I'm not going to debate what causing the changes, but we know that the environment is changing. And as things warm, and we notice that we've had warmer summers, that creates habitat for hydrilla. So hydrilla, which historically this may have been its northern limits for the monoecious form. Now we're seeing it potentially, we're becoming a much better habitat for it, and it's starting to extend up into Canada and stuff like that. The other thing we want to do here is significantly reduce that risk of hydrilla spreading, and if, like I said, if we can eradicate. But I'll second all the speakers before this. I want to thank all of the partners. They're integral to this project. It's great to work with such a good, diverse group of people who have been working on this for an awful long time. Very passionate about this project and protection of this lake and this incredible resource we have here. It's also, it's great to see the feds, the state, the locals all working hand in hand. We contact each other all the time. We have monthly meetings. We keep in contact, and it's just a great project to be able to do. And it's nice to see funding starting to dry up in one area. You have others that are starting to scramble around trying to get funding sources and trying to pick up where that potentially there's going to be a gap, so that we don't have this loss of the controls that we have, the gains we've already made this year. So management options that you usually have for aquatic plants. We have biocontrols, that's been mentioned. So you can have an insect, something that actually preys directly on that, eats that particular plant, selected biocontrols. We don't really have that option available to us for monoecious hydrilla. There is for dioecious hydrilla. The main difference between those two plants, the dioecious is a more tropical form. It actually grows year round. So there's above the sediment surface, there's plant biomass there year round. So that's why a lot of applications of dioecious hydrilla actually occur in the wintertime, as opposed to the summertime, when it's actually growing. It's slower growing, so there'll be some applications in the wintertime. Here, with the monoecious form, it actually dies completely back to those potato-like tubers, to the seeds, or to what they call turions, which are those vegetative structures that are from the above ground vegetation. So that is what dies back. So there's really not much there for another animal that may feed on that particular plant to actually survive throughout the. So the selective biocontrols that we have for some of the plants that we have up here, we kind of lose that. Grass carp in a closed system in a pond. We'd sterile grass carp. naturally reproducing, which we're dealing with out in Lake Erie, can be used. Okay, but in an open system like we have here, with a lake that's 66 square miles in size, or roughly 42,500 acres in size, that's an enormous area to have to try to stock enough grass carp to have any impact on the hydrilla population. The other thing is they're not selected. They don't just look for hydrilla and eat hydrilla. They eat other more nutritious or easier stuff to eat, like valsinaria, like some of the other plants that are really, really good habitat for fish. And they will move out from usually can't keep them just in one grade. So that's not a really good option, like it is in certain systems in that. Mechanical harvesting. You can harvest for that. Unfortunately, with hydrilla, what ends up happening is because it grows so rapidly during the growing season, and because it vegetatively propagates from these little clippings, you go through with a harvester, which creates all these little bits, and it pulls some of those up, and then a lot of it spreads out all over the bottom. Now you just took an area that may have been a little bit of hydrilla and some other native plants that are there that are actually kind of competing with it, and you just made it much easier for hydrilla to reestablish and become even more dominant in that area. So harvesting actually, not only doesn't it work, it actually promotes the spread and the expansion of hydrilla to get to that monoculture where it can become a real problem. They tried diver suction dredging, I believe, when they were doing the inlet. They tried to do some of that work. Very time intensive, wasn't very effective because hydrilla is very easily fractured into these small little fragments. It doesn't hold together very well. You grab on the hydrilla, it just breaks into pieces. So it's ideal for doing what it does, which is to survive. It's not really good for helping us try to control. Environmental extremes. The tubers that it produces to vegetatively to survive and to get to the next year, those tubers can last up to 10 years. You can freeze them, does not affect them at all. You can heat them and denature the proteins and everything in there, and you can kill them. The problem with that is they're in the sediment. So you have to heat the sediment up high enough in order to do that. You can do that in a canal, in an area where you can control the water levels. We can't do that in the lake. So there are some things that we could play with, not an option. So really what it comes down to is you either do nothing, in which case now you let hydrilla go uncontrolled, and it will outcompete many of the areas that we have here. It'll grow in areas that other plants won't, which is how it gets established. Then it starts to spread into areas which are the better quality areas and start edging those plants out. That's why a lot of times you'll first see it in with other plants. You come back a year or two later, now you have it basically, it is nothing but hydrilla. So then we're left with the most cost-effective and most effective option that we have currently, which is to use use herbicides that the EPA and the state has deemed as being safe and can be utilized to go after hydrilla. The advantage that we have, or what we try to do when we're doing this, is each type of plant has a different susceptibility to certain herbicides. In the case with us, we generally use fluridone here because we are dealing with potable water intakes around some of the areas or within proximity of where we're treating, and that's one of the herbicides that we are allowed to actually use as long as we use it at low levels. And hydrilla happens to be very, actually responds to that particular herbicide at lower levels, where other native plants don't as much. So some native plants are effective, but they're not as effective as much as hydrilla. Elodea is not effective as much as hydrilla at the same level of concentration. Milfoil, absolutely, but another non-native, and not as abundant in the lake proper as it used to be 10 years ago when we started this. Your valsinaria, a lot of your native plants, your your pond weeds, many of those actually have a little bit better tolerance to fluridone at the levels that we apply at than Hydrilla. So we're trying to be as selective as possible when we are having to use these herbicides, keeping them at as low levels as possible to reduce as much of the impact on the other native plants, because we recognize plants are good for fish habitat, for waterfowl habitat, for a lot of things. And we want those native plants, a good, healthy native plant population actually helps suppress Hydrilla, keeps it from being able to spread very easily. Go ahead, next. So I wanted to take a little bit of time to explain why we do what we do with the herbicides. Not that we just bypass all the other options. It's really the only good option we have. We can use benthic barriers in certain cases, and they are used. They're very time intensive to use, labor intensive. They also can only be used in very shallow water. Hydrilla will grow out to 25 feet easily here. We catch it at 25-foot depths. I can't send someone else to put a benthic barrier unless they're doing diving. Once you add a diver to it, now costs go up and the labor and the amount of labor to deal with that goes up. So two of the herbicides that we generally use when we're going to be doing any applications, larger, and when I say larger, in the grand scheme of things, these are small treatments that we're doing here, are we use fluridone pellets because we want the pellets actually allow the herbicide to be along the bottom, and fluridone works as a systemic herbicide. Basically what it does is it interrupts the plant's ability to create photosynthetic pigments. And so over time, the plant no longer has chlorophyll in it. And so they start to bleach out and they can no longer produce the energy they need in order to survive. They end up dying. That process takes 45 to 90 days. So you have to apply the herbicide at a level that's going to interrupt that chain so that basically what it does is there's a protective enzyme, a carotenoid that protects the photosynthetic pigment. Or the chlorophyll actually starts to break down and it can't. So it just starts to bleach out. That takes 45 to 90 days to happen. So anything that we're putting out there is low levels and it takes a long time for it to actually happen. So it's not like we use the other one, which we use for very small spot treatments. These are for 5 acres or less, very small. We have a patch that's either not responding in our treatment area. We want to make sure that we treat that patch and it's pretty much solid hydrilla, so we tighten in and we just apply the copper right to that area, or it's an area outside of our treatment area that we found a patch and we want to get a knock on that patch. So we can use that. It's okay. So this one is a contact herbicide. This one does kill the plant very quickly within a week or something. This one is also much more broad spectrum. This impacts algae and all that. It also is to the aquatic organisms at the levels we're using it. They're considered safe levels, and also the way we apply it. But you have to be careful when you're using copper for an extended period of time because it can become it doesn't break down in the environment like fluridone does. Fluridone within 20 to 50 days breaks down. Breaks down from sunlight, it breaks down from microbial action. The harpoon, on the other hand, that doesn't break down. So it can stay there, out there. And if you were to use it for an extent for long periods of time, you can start to see the levels of as you can see here, we apply at a maximum of 20 parts per billion. So we are literally applying at less than half of what is allowed for safe drinking. So we are applying this herbicide at low enough levels that we don't have that. We test it weekly. It's posted on the Department of Health webpage. We notify every property owner and we put signs up at all the public access areas to the areas that we're applying the herbicide as we're required to by law in order to ensure that People are aware that if it exceeds 1 part per billion, then there are some restrictions with regards to watering your tomato plants or your pepper plants or stuff like that. So there are some things that if it exceeds 5 parts per billion, generally is where you start to worry about that. But at least you can look on the website, you can contact us, we'll let you know if you may want to hold off on it. But generally speaking, we apply at very low levels, and based on our water testing, we're trying to keep the fluridone levels at a 3 to 5 parts per billion level. That's what we consider a concentration that's lethal to maintain for the hydrilla, but it's low enough for everybody else. Usually, most of our tests that we do are less than 1 part per billion. So it's difficult to kind of hold that in that area. And those pellets are slower release. So you'll see a lot of times the first few weeks, you don't get that much concentration, and then we hit a really good time, and then it tails off at the end after we're done. We do that water testing throughout the entire season and put that up onto the Department of Health website. Go ahead. Okay, before I jump into actually our treatments and what we did, are there any questions with regards to why we're using herbicides, what herbicides we use, any questions like that? Yeah.
Yeah, maybe to get to this, but what sort of metrics do you use to gauge the efficacy? So, yeah,
So
the hydrilla before and after, and say, well,
yeah,
by X amount.
yeah. So, and I'll show you some of those results.
Okay.
So what we're doing not only is we're doing the water testing, which is giving us an idea. We have weekly water testing we do to make sure we can check those levels. And so before we reapply, we make sure that our levels aren't getting up high enough in which we want to hold off and wait for another week to apply, because we don't want to go over that 20 part per billion time, which, like I said, normally we're almost always less than 10. Very rarely are we over 5 with that, especially this past year. So we'll do that. So we monitor the water temp, and then we actually go out and do those rake tosses and point intercepts, and we look at the plants. We look to see, we throw 3,000 different points in Aurora, and then in Ithaca, another 400, almost 500 down there. We go out right at the beginning of the year when we first start applying, middle of the year, and then we do it at the end of the year after that to see what survived and what we have to worry about in the upcoming year. Because if they have survived and they look healthy enough, they're potentially going to put down tubers, and then we have to worry about treating that patch or that area. Again, if we don't see them, that doesn't mean we don't apply there that next year. It just means that we did a good application in that area, and we now have to exhaust the tubers that are there, which, generally speaking, if you get a patch, a plant goes into, or you get a patch started, in order to totally eradicate that tuber bank, it's 10 years. 10 years of application to be eradicated. Because those tubers do not all sprout the next year. A bunch of them do, and then some wait for the next year, and a few more wait for the next year. So in order to get every last one, you can pretty much think about doing that for the next 10 years or so. So, and that's where it gets very hard, like Roxy's talking about. Initially, you see lots of hydrilla in a big patch. You go out and you apply herbicide on it, and you knock it back. And then the next year, there's less. And the next year, there's even less. It's when you get down to this, like we refer to it as the whack-a-mole stage, which is where you have the patch that used to be there, and all of a sudden, oh, there's a little patch. There's a little patch that just popped up, or a little plant that popped up, and another one that popped up over here. We apply there because we're trying to kill those as well. Because if we don't do that, now they put down tubers and they just roll. You just lost some of all that gain progress you just got. You now lose that because now you have plants that just put new tubers, and those tubers potentially will sprout over the next 10 years. It's one of the difficulties we have with treating hydrilla. Okay.
There's one
Yeah.
question on chat that
Sure.
says, how about forever chemicals?
PFAS. So I will give an answer that is US EPA in New York State tests these herbicides, and they select which ones are safe to use at the levels that they're going to use. To my knowledge at this point, and I have a rep from CPRO that can verify this, but I do not believe that US EPA has identified any PFAS in sonar or the herbicides that we use at this point. That includes the.
It's been tested?
Yes.
Yes.
Well, the subtlety here is, of course, the definition, right? So I think what PFAS is defined as, I mean, according to the US EPA, is that the active.
Correct.
Not according to the definition that New York State has used, some laws, but also many countries use the OECD definition. The active ingredient would be peptide. Right, so that's the.
And that, like I said, at this particular point in time, our stance is right, exactly. That is outside our purview. My purview. I am no expert. I'm not a chemist. It's up to US EPA and New York State to make those determinations as to whether they want to use that more broader definition and if they want restrictions on that, because that broader definition, as I understand it, those chemicals aren't necessarily as detrimental as the more restricted one that has the larger, more limited.
As I understand it, the issue here is that it's a rapidly evolving crux of unknowns as to the case. For example, is that, and one question I had on that issue is that you say the lifetime is 20 days. It can often give the impression that, you know, the molecule just sort of vaporizes into nothing at the end of that lifetime. But of course, there's a decay byproduct.
Correct.
One question.
Two main ones at least.
And for that, I mean, you know, the lifetime issue, like lifetime in sediments versus water, and the old sediment where little light goes. I mean, there are studies that, and lots of information online. I mean, the lifetime can be many months up to a year, right? I mean, there are competition. So, two questions would be: I mean, what monitoring have we done of you know, residual concentrations, sediment also, where light doesn't reach at longer scales, and of the decay byproducts, right? Which I think are a separate matter from the active.
Correct. They are. And as I understand it right now, those two byproducts, one of them, the one that some have shown to have some detrimental impacts, have only been identified in the lab. They've never been found in any study that's looked at the breakdown of fluridone in the field. It's never been found in the field at the 150 parts per billion. So at the max label rate, you are allowed to apply it. Remember, we're at 20 for a seasonal total of up to 150. That's our max. At that level, if they were to put 150 out there and have that breakdown, they have not found that 110. Have not found that they have found it in the lab, never in the field. To this point. So as with any regulatory type thing, it's always evolving. There always are going to be changes. And if that happens, and we are told that all of a sudden now we can't use sonar H4C, which we are allowed to right now, and there's another one, then we would have to adjust our management plans.
Have we measured that anywhere in Cayuga Lake, or you're referencing some
I'm referencing studies, all the studies that have been done.
And the difficulty in interpreting these statements is that, you know, if very few studies have been done, or they haven't looked at the sensitivity limit that would apply to concentrations of parts per billion, then it's hard to know if that means that they're not actually there, or what. The question of if we've looked in a sense. And I can't really answer that.
Yeah,
I mean, it sounds like we haven't looked empirically.
Again, there's not been that. I just know that based on the studies that have been done to this point to look at that. And again, any of the herbicides that have a label on them that are certified by the state and by US EPA for us to use in aquatic realm have been through extensive testing process from the UK as well as New York State. Because there's an additional, that's the thing in New York, we have an additional catch where there are US EPA approved herbicides, and then they put additional, they have their own label, so they'll put additional restrictions on that probably use it in states. And we follow all of those labels. So yeah, go ahead, Joe.
Can you share the data that is on PFAS analysis for the technical grade material? Comments on that?
Yeah, go ahead.
Sure. Well, no, not, I mean, I can't. Not now. I would say it's in a written form so that we can actually.
Well, I have resources at the end of this, and at those resources, at those websites, you'll be able to find that.
Okay, good.
I know John is also potentially may be able to provide some additional resources.
That'd be great.
Okay. Yeah.
I had a question, you know, obviously it's a watershed, so that's pretty important. I'm curious though, since these chemicals have started being applied.
Yep.
Have there been changes to the testing that's done on the water, like to update the testing? Is it testing when they first water testing for all these additional things that are being added to the lake? All the additional herbicides and things, or is it the same old that isn't really testing for copper and the heavy metals that are being added to the lake?
So anytime that we do our application of an herbicide in the area where we're applying it, we do water testing for that particular herbicide, whether it's sonar, that's a weekly testing we do.
Right.
As per the Department of Health in the state. And then if it's copper, I do a control outside of the area that we've treated and a treatment in to compare the copper levels to see if it's background levels or if we're starting to have increase. And then that would be compared against the water quality standard that the state gives as to whether or not that we are exceeding that or having an issue. And that's what we follow. Our rules are as a state and make sure that we're not exceeding any water quality or water standards. So.
Can I ask one more? This is in the chat. How much funding is allocated for eradication as compared to research into biological controls?
Honestly, I don't know. I don't have those numbers. One of our colleagues who used to work on this project, my career, runs the aquatic plant control program for these states. And he might be able to provide a little bit more information. So I might be able to provide some information on that, but right offhand, that's above my pay scale. So.
You know, I think there might be confusion because your funding comes under a line for research.
Correct.
That might be confusing, folks.
Okay, so yeah, that's the other thing, and I'm glad you said that because I meant to make that comment at the beginning. The Army Corps themselves, we actually have an authority given to us from Congress that said we're going to give you the ability to be able to control to treat aquatic invasive plants. So we have an aquatic plant control program. Within that program, we have a component that allows us to do research and development work. And that's what the authority that we use in order to do this work here on Cayuga Lake. And this is why I say here, these are large, we think of them as being larger projects, but in the big scheme of things, I talked to the folks in Florida and they chuckle because they're treating thousands of acres with one application. And we treat a total of 150 acres between Aurora and that's it. So we're talking about thousands of acres in one application where they're managing versus us only looking at 100, 150. But the research and development piece that we're doing this under is these lake systems where we're dealing with are high mixing areas. The canal, high mixing area. Very difficult usually to treat traditionally with herbicide or to treat with anything. So they asked us to kind of come in and to look at novel ways to be able to either apply the herbicides that are USDA and New York State approved. And remember, as we all know, if you do max legal rate, everybody knows you can kill plants. We're trying to find the sweet spot, and that's where the R&D comes in, where we have effective treatment on hydrilla, minimize impacts to the native plants, and do that as cheaply and cost effective as possible. Because that in reality is what these more long-term management projects that are going to hand it off to the locals are going to be able to do.
To the state.
Remember, I'm Fed, so Fed would be state or local, okay? So in essence, what we're trying to do is to take and find different ways to be able to apply this herbicide, potentially. And when we're talking about different ways, well-timed applications. Hydrilla, when it first comes out of the ground and sprouts, at that time, that tuber will never sprout again. It's dead. Okay, so whatever happens after that, that tuber will never sprout ever again. That usually happens here in Cayuga, late June, early July. It's probably going to start to shift earlier in June as the water temperatures in the springtime get warmer. We do find it sprouting a little earlier in the inlet where it's warmer temperature, so it will sprout a little bit earlier. So sometimes we use a little bit earlier time there. But sometimes the turbidity actually suppresses the growth too, so we're kind of using the same ones because we usually have better clarity in the lake.
But just if I could jump in, related to this question of the research components, the reports did mention this, like, part of the purpose was, I mean, as stated, and I understand it's something that funding category, but a demonstration of technology developed under the aquatic plant street research, whatever. Well, what is that technology then? Is it evaluating fluoride concentrations, or is the H4C formulation something that was developed as a collaboration with SE Pro? Is that the technology, or what technology is that?
The technology that we're referring to sometimes is testing new herbicides and that in other areas of the country where they are allowed to use certain ones. In this project, in particular, it is using existing herbicides and existing technologies, but doing novel ways in which we apply them to try to maximize their effectiveness against hydro. Yep.
So you talked about the whack-a-mole issue, and you know it's an interesting problem because here you're trying to maximize the effectiveness of the herbicide while using minimal amount, and then at the same time you're having some success, but then wait too long, something pops up over here, and so it almost seems as if it is a long-term management strategy that I guess we're embarking on. And then has there been success elsewhere in the country where actually edited hydrilla from a water body?
Get back a while.
Yeah, I know there's been some, but
river, another part that it's very small.
Yeah,
it gets to the water body this size.
Okay,
it could be.
it's the earlier in on the infestation, the better chance you have.
Yeah,
the more control you have over the conditions, I think. As
well, and I'm just trying to make sure people understand expectations here, because this is a long-term plan we're embarking.
So that's kind of some of the discussions we've been having amongst the group is, are we still at the eradication phase, and that's why if you saw my goal was where we can eradicate it in new patches and that, try to protect certain areas. There may be to the point now where it's dispersed enough in which we have to look at this as a more long-term. We deal with Tuning Lake on the Ohio-Pennsylvania border. We work with Pennsylvania, DCNR there. And they had such a large infestation before they were able to get the funding to start doing the applications. They're already in that. They've done a really, really great job there of knocking back the hydrilla from high priority areas, which would be boat launches and at the dam where it could potentially escape from the lake and get down to the next dammed reservoir, which happens to be a core lake. And so what they're doing is they're making great strides in certain areas, but they also recognize that sometimes they may have an area pop up that they think they can eradicate. That's going to draw from an area that they're doing management on. Well, they may be willing to let one area that's lower priority sit for a year, recognizing there's going to be some plants that put tubers down. That means we're going to have to treat that longer. But we're going to go after this, hit this. So those are the types of strategies that they've been having because they're farther down the road than we've.
It's a trade-off of resources today.
Yeah.
I love that we love asking Rich questions, but I want to let him finish his presentation, and then we'll do more questions. So like hold those in your mind. Emily has a short talk as well, and then we'll keep on.
Yeah, I apologize.
That's okay.
She's sitting here trying to get her stuff done. As you can tell, I was a professor for a while, so I like that. So I'm going to quickly go through this. This is just showing you what we did this past year, and kind of what's in educating us on what we're going to try to do. So go ahead, Aurora. So I'm going to run through these pretty quickly. So like I said, we treat about 150 acres. Aurora started out down here. This is Wells College Bay. Right here is the Wells College dock, and here's the bay. So we still treat a small little area right there in front of the water treatment plant because last year we found a few patches. We haven't treated there in a couple of years. We do monitor that. We saw some patches come back. We didn't want them to get reestablished, so we want to back it up. We have had some patches here for several years and haven't seen anything, so we decided with the limited funds that we have, max amount of funds that we have, prioritize treatment of hydrilla to the north to protect and make sure we don't let hydrilla get into that northern area. As Sam mentioned, the southern part of the lake and the northern part of the lake, that's where the majority of the littoral zone is. So it gets really shallow. It will be much harder to treat up there doing that, and it also then puts it to the point where they can escape to the canal. So this is Mackenzie Childs, for anybody that knows the area pretty well there. This is Miss Mackenzie Childs, so we affectionately call this the Cove, Mackenzie Childs Cove. This is a shallow, little bit more broader area here that's shallow before it drops off. And when I say shallow, I mean out to 20, 25 feet, then it drops down into 50, 60, and continues on. Go ahead. So we did, this past year, we did in these areas, kind of modified our application, recognizing we were trying to maximize the amount of area we were treating, worked with our partners, come up with a little bit different treatment plan than we normally do. Instead of doing 10 weeks of applications with a max 150 parts per billion over that 10 weeks, we decided to go with 7 weekly treatments at 20 parts per billion in the hopes that if we well-timed this right as the plants were starting to come out of the sediment, that's when they're actively growing and they're going to take on most of the sonar. If they're damaged in that smaller phase, and we weaken them and damage them enough, we don't have to do as long of a treatment. We can get away with doing this, and then we end up only doing 140 parts per billion for the season. So, so we ended up. Here's our monitoring areas. We always do a half mile north of where we're applying. DEC was kind enough because we used to treat just here, and then we started treating down here, and then all of a sudden we started noticing patches moving this way. These patches are about the size of a tire. That's it. So we're not talking massive patches. They're just small patches, but they string together. And so you can't just treat an individual patch. You got to kind of treat an area that has them in it. And then DEC has taken over treatment in this lower section. And this is Long Point State Park. And then we do a half mile below there. So we take care of the water sampling, all the fluridone applications that are coming at what we affectionately would call Aurora, but now it goes all the way from Union Springs down to Long Point. Go ahead. On that weekly testing, as I said, we didn't have anything really exceed 5 parts per billion. And we always make sure we test finished water at the water plant, and it never exceeded a half part per billion. So not only are we allowed up to 50 parts per billion, it never went above 0.5 parts per billion in the actual water, and that's the lower detection one. So it was not detectable basically. So what we did is we throw 3,000 plus rate tosses with DEC, Finger Lakes PRISM, and U.S. Fish and Wildlife. We all get together and we go out and actually spend a week Fortunately, in October, we spent it bobbing around in really nasty weather, but we managed to throw about 3,060 rakes or so in this area, and then identify any of the patches, any of the points where we found hydrilla. The red is showing what we found in October. The yellow is showing throughout the entire year. We've sampled 3 different times of the year. Like I said, at the beginning of the treatment in early July, in mid-August, and then again in early October. So this is all the hydrilla we saw, and then these are the ones that we found in October. So what you can see is we had pretty good treatment in these areas, especially we oftentimes see patches right along the edge of our treatment. Plants are there, but they're not looking that great. This year, though, these plants in this area actually looked really healthy. So we did 7 weeks instead of 10 weeks, so we shortened the time period in which we were applying it. And what happened this year was the months of August, September were some of the driest they've had on record, and also were really warm. So although we weakened the plant, and we did see very effective treatment when we were out there in August, by the time we went back in October, the plants had recovered, reproduced their photosynthetic pigments, and were starting to grow again. Now, the plants were only that tall, as opposed to being much taller than me, which is what they would have been had we not applied it. But they still were doing that, and there's a possibility with the warm weather and the warm temps that they will grow, continue to grow until the temperature drops below about 62 degrees water temp at the bottom. So in probably the late October, they will continue. So next year, we're going to concentrate our efforts right in here. We do have some hits here, so we're probably going to play around with doing some stuff here. This is really going to concentrate our efforts. We had some patches originally in Ellis Point, and we'll look and talk about possibly pulling some of that back and saving that and utilizing that in here. And we've already talked about the fact what we're going to probably do this upcoming year is we're going to start with that 20 part per billion for the first 2 to 3 treatments. And then we're going to back off and go with about a half treatment for the next few weeks to try to extend that time period, that tail of the fluridone, and then go back and try to get that so that we can extend the months of application from that late June, early July, all the way to the end of August. We can get it to the end of August, and usually the fluridone tail will extend through at least a good part of September, and then the plants won't be able to have as long of a time to recover. If we get weather similar to what we had this year, we could get crazy storm events and all kinds of stuff next year too. So. Okay, next slide. I think the key thing to remember here, when Bob went out, 50% of his points in that 30-acre area were going to be hydrilla. It's growing to the top. We are seeing hydrilla spread out over 75 points throughout the year. That still amounts to over the 7,800 rakes that we threw, or samples we took. We only caught it at 75 places, so a little over 1.3% coverage, which isn't nearly as much as what we would have expected if we continued to allow that to go. So a lot of the impacts that we could have had because of the applications that we've done have kept the hydrilla at low levels and are not allowing it to recover as much. This is just showing I added the hydrilla points that the DEC covers. And so we've got through some of their treatment areas, and then we have some additional points that are down here that are not on.
Yeah, so you said some of the treatments that are keeping the hydrilla down at low levels, and that's hard to gauge from just one year's reports. I mean, what you need are comparisons of how they've evolved over time, and I think average. Yeah. But to make a claim like that, I mean, you always in science need a control, right? I mean, to know that it was your intervention that caused that relative, whatever the complicated dynamics of nature otherwise. So how do you know that it's the treatment that regulated it? What's the control?
So the control, we don't have an area that we know is with hydrilla that we do not treat here. But we can look to other similarly situated areas and that we don't treat, but that hydrilla is doing very, very well.
What are
So that's. Oh, man.
If I may,
Yeah.
you were asked to hold up questions.
Yeah.
After you finish, can we do that? I have questions too.
Yeah,
so
yeah. Let's do that. Let me finish so Emily can do that and we can get on to a few more questions. Go ahead. Next one. All right, Ithaca. Ithaca is a much better story. You saw Roxy, huge area right by the co-op, by the farm stand. Now, go ahead. So historically, this is the farm stand area and Cascadilla Creek. Historically, there was a bunch found in Fall Creek here. This is Stewart Park. We had a large patch up here in 2017, 2018. They found Bob found some patches coming back. This is the area that was originally treated back in 2011, 2012. And what we have slowly done over the last 5 years of applications is been able to pull this application back. We were doing a drip in Fall Creek. We're no longer doing that. Golf Course Lagoon was always a shallow area, really warm, that had a ton of hydrilla in. We haven't seen hydrilla there in the past several years. So we think we've got a pretty good control there. No hydrilla found out in these areas either. These were more protective measures in case there were fragments in here. And then concentrating on high profile areas, the boat launch at Tremont State Park and the marina at Cascadilla Creek. And then this is Cornell Crew Bay. And we always seem to have fragments floating around here with the amount of traffic, boat traffic that goes on there. And then this past year, we found at the tip right here, which is the Boatyard Grill, we found some patches there. And after this year, after the applications we had, go ahead and scroll through the next 4 slides. We'll get to the end. Yep, keep going. Let's see. One more. Water treatment, water sampling, never found anything above 0.5, just like the other water plant. Go ahead. Last thing, that is the only area where we find hydrilla right now. Cornell Crew Bay still is an area that we're going to look to treat. So what's probably going to happen this year is we're going to pull this back and we're going to concentrate right Cayuga. So we have done exactly what we tried to do, which is we can knock it back. We're going to shrink the area, we can hopefully eradicate it. Ithaca is one of the few areas where we're actually able to make a significant back.
Okay, thank you, Rich. One question per chief while we switch the slide.
My question is this: state resources in the long term, long-term project. My question is, what are you all doing to the softwares and GCC plan long term? Do you have a long-term treatment plan? Do you have a long-term funding plan? What's going to happen?
As far as funding, typically we get a decent number of our funding from GLRI, or Great Lakes Restoration Initiative. And we currently don't know, we apply every year for grant or money. And so we don't always know exactly what we're into, but that's typically where we're at.
Is the treatment plan year to year?
Typically, so we kind of adjust our treatment plan based on what we find the year before. So kind of as Rich said, where we're not finding stuff in certain areas for maybe a couple of years, then we'll pull back from that area and either focus on another area or eventually pull back.
Well, that I understand, but that's an adjustment for what? My question is, do you have a plan that then is adjusted to and modified based upon the situation? Or is it each year you look at what you've got and make a decision about what to do? It's not really a plan, it's just reaction.
Um,
It's okay if that's what it is. I'm not trying to.
yeah, I guess it is, we do have, you know, the, Cayuga Lake Hydrilla Management Plan, and within that, it does allow for adjustments to be made. So, you know, that answer.
Nice, sneaking in some questions before we talk.
Yes.
So, Emily, you can take your time.
Okay, all right, yeah, I mean, I think a couple of things that I was gonna talk about were already covered, so I should take so long. So, just really quick, I'm Emily Denzinger. I am the Aquatic Invasive Species Coordinator for DEC Region 7, which covers pretty much all of Cayuga Lake, and then we have that little sliver that is also in Seneca County, which is Region 8, but like luckily I get to cover that as well. So I pretty much cover all of Cayuga Lake, all this aquatic invasive species happening there. So go ahead. Okay. So just real quick, what I'm gonna cover, just go over our field season summary, some of the management methods that we do use in New York State, which Rich also went over. So really talk about that too much, and then some of the other hydrilla management projects in New York State, and then just a little more information about what DEC did on the lake. I work with 4 seasonal technicians, or I have 2 interns and then 2 other seasonal technicians, and we are out on the water pretty much every day from end of May to the end of October. And this year we did over 6,000 monitoring points on Cayuga Lake. And we also, you know, we did about another 2,000 in other parts throughout the region, so we are pretty busy. And we'll usually do about 2 rig tosses per point or more if warranted. And then on Cayuga Lake, we monitored over 1,000 acres, and most of that we monitored twice. So we go back and we'll do a monitoring event, usually earlier in the season, so May through June, and then we'll come back and we'll do a monitoring event later in the season just to see if there have been any what kind of changes in vegetation there have been, and also like a pre and post treatment monitoring as well for any of our sites. So just really quickly, these are our monitoring locations. So just kind of listed from north to south. The sites with the red circles around them, those are sites that detected hydrilla at this year. Most of those were at previously known detection sites or in the vicinity of sites that had history of hydrilla. So no shocks and surprises this year, which is good news. That little star down at the bottom, I know we do have hydrilla down in the Cayuga Inlet. We were just at Allen Truman Park, just monitoring in that area. So that's down there. Excellent. And then really quickly, just to kind of cover like how do we monitor, what are we out there doing for we go out, we lay a grid over our area of interest. So we'll use a 50-meter grid for any early detection sites. So those would be areas that haven't had any previous hydrilla detections or management efforts in those areas. And then we'll do a 25-meter or a tighter grid for no hydrilla locations. So areas that we're currently managing or areas that they're made past management events. We'll do at least 2 rig tosses at point on either side of the boat, or more if needed. And we'll kind of total up, or we'll take a density of our total vegetation that we bring up on the rig, and then individual species density as well. And we record that on a scale of 0 from 0 to 4, so 0 being no vegetation, and then 4 being super dense, hard to bring the rig in the boat. And then we'll also take some sonar and biobase data as well. So this is just a quick representation of what we're looking for when we're out on the boat, and what we're recording. So if you hear us refer to like, oh, it's a trace hydrilla, it's usually just like one little sprig of hydrilla. If it's dense, it's usually the whole entire rig. It's hard to put into the boat, and guess for those who are not familiar, you know, the rig toss is this. We take two like hard metal rigs, we zip tie them together, toss them off the boat, kind of let them drag for a little bit, and it'll pull up all the vegetation, and that's kind of you know what we're finding. Just some more photos from the season. That's a lot of plants on that one. Yeah. That one for sure, that's all Hydrilla. I'm not 100% sure what this is. This is taken this season before I started, but. We'll also record biobase and sonar. So that is that blue and that up there. So biobase or the bio volume is the percent of the water column that is taken up by vegetation. So, you know, the water's 10 feet and there's 7 feet of vegetation, then that would be 0.7 or 70% bio volume. And we kind of do this, you know, we're taking points. We cannot record data at every single inch of the lake. So the sonar just kind of helps supplement a little bit of what we're doing. That's just kind of covering like what we're doing, what we're out reporting when we're in the field. So Rich covered a lot of these, so just our management options. So, you know, no management, physical control, which there's different methods in there, biological or chemical. And then if we do use any of those in combination, we like to call that integrated management. So no management, you know, it is an option. Initially, it's pretty low cost, but then you know you do end up incurring a lot of those costs, those economic costs of what happens when the hydrilla spreads, when these mats are super dense and you can't navigate, you can't fish, limits recreational opportunities, as well as having to really increase efforts in outreach and education to prevent hydrilla spread into other water bodies. So those watercraft stewards that Sam works with in Cayuga Lake FL-PRISM, you know, they're doing a lot more. And you know, it does put the other water bodies around Cayuga Lake at risk for hydrilla spread, or more higher risk, I guess. There's always still that risk there, but 19 that risk. So next one. Physical control, Rich kind of covered this, so you can, just for the sake of time. Another physical control, which I don't think Army Corps went over, was dredging. It can be very successful in like very small, isolated areas. For an example, Finger Lakes FL-PRISM, they helped fund the dredging project in 2019 in a small marina in Cayuga Lake, and they have yet to have any future hydrilla detections. Now, this, you know, if anyone has ever done any dredging or been involved in any dredging projects, it needs to be a really pretty relatively small area. Dredging all of Cayuga Lake is not an option. So, you know, again, this is just good for small, isolated areas, and this is like an early detection, rapid response for hydrilla. Next one. Biological, so again, already kind of covered grass carp. Some people use those. They can be used to control just aquatic vegetation in general, and like smaller, isolated ponds that don't have any connectivity to other water bodies. And then right now there are no other current biological control agents to control hydrilla. And then chemical. So this is again typically the option that we do use for hydrilla control in Cayuga Lake and at some other sites in the state. It is ideal again for large scale populations where some of those other more targeted options might not be useful. And then we are required, you know, even though we are a state agency, we still have to apply for permits. We have to permit ourselves. You know, we don't have the ability to just go out and apply herbicide, even though yes, we are the EC, we still have to apply. We have to submit a permit to the risk management for the pesticides folks with the DTC that goes in for internal review. as well. So just like if anyone else was going to apply for the permit, we go through the same process. And then same Article 24 permit if we're going to do it. So then just a little bit of the hydrilla management in New York State. So again, just a general timeline of all the hydrilla that's been detected in New York. So first discovered in 2008 downstate, and then the first discovery in Cayuga Lake in 2011. And then just as time's gone on, we've more. Again, a lot of this, not a lot, a decent amount has been found in smaller private ponds. But may not necessarily have the resources or the time to pull in. So this is just a quick distribution map from, I pulled the data from invasive. It's a database that's widely available to the public. So anyone hop on there and take a look at this information, you can. So DEC this year, as Rich mentioned, was responsible for doing management in the South Aurora area. So Army Corps, they were up here, we were down here. We used sonar H4C on 127 acres of surface acres. We began our treatments about second week of July. Those went for 10 weeks until mid-September, and then we do collect water samples after each treatment to ensure that we're, hitting the levels that we want to meet with our herbicide concentration, and that we are use restrictions that might be in place. So for the next one, this is a lot of numbers. But the most important thing is these are concentrations that we apply herbicide at every for each week. So we started out with 20 the first 2 weeks, and then we went down to 13.5 parts per billion the second week, and up 100 throughout the whole thing. And so the label we're limited to 150, so we were below that. These are our sample locations that we take samples at, and then we also do have samples half a mile north and 4 miles south. We also did some management at Sheldrake and Wyers Point. So we based our 2024 treatment off of our 2023 hydrilla detections. So this is the map from 2023. We did some pre-treatment surveys in June, kind of through July. So the Sheldrake area, we monitor it's about 15,000 points that we monitor. So it usually takes us a good 2.5 weeks to get through all of that. We're doing that at a 25-meter grid. So it takes us a long time to get through it. So that's why it's a little longer. And then we did another pre-treatment monitoring event in August, right before we did our treatment, just to really narrow down the area we're flying in. And then a post-treatment in September through October. So again, this is just kind of a map of what that looks like. These tiny little polygons, those were our treatment areas. We treated a total of 2.3 acres using granular, so just under 200 pounds total were used. We also did a couple of very small spot treatments in Finger Lakes Marine Service, again, based off of our surveys from last year. These were super small. Each point was about 1,000 square feet circular. Again, we used about 3.5 pounds just to do a spot treatment in those areas. And we did those in mid-August. Same with Myers Park. We also did some treatments there based on some detections from the year before. Same thing, we just did a couple of smaller 1,000-foot treatment areas, and we used a little over 5 pounds total. And again, using granular, which is and then these are just some other hydrilla control projects going on in New York State. So some of the bigger ones are, Erie Canal, Niagara River. Another one, Croton on Hudson downstate, dredging, which I mentioned earlier, that happened a couple years ago. That's still being monitored, so it's kind of considered still kind of ongoing, even though we did bulk of our management already, or Finger Lakes PRISM did the management. And then biological control, just in a private pond, and then a few areas where they're doing a combination of different management efforts, but they are switching to just doing herbicide in those areas. So our post-treatment monitoring. So this is what Rich mentioned, those southern detection points that we had. So these two we found in October, and then this one we did find a little bit earlier during our August monitoring. And we decided to go out just for one day. I was like, oh, we'll just go south of our regular monitoring grid and see if we see anything. And we found hydrilla. So yes. Next one. This is Sheldrake. So this was from 2023. This is currently from this year. So unfortunately, we did see an expansion of hydrilla. We did find some further up here and further south. So this past year, I will say, so these yellow points, this is what we found in July and August when we did our pre-treatment survey. And so we're like, oh, we're good. We're going to be able to treat all of that. That was in our treatment area. And then when we came back in October, we did end up put our next one. And Myers Park, Lansing, Ladoga, kind of a similar story. Again, we did find, in our earlier pre-treatment surveys, we were finding hydrilla in the areas that we had found in the year before. We treated it, and then we did end up finding a little bit more outside of our original. So, but that was, I think that's it. I think